in the third part of this session, I'm going to talk about the anatomy and the basic structure of the visual system. Just give you a sense about the basic structure of this complex system. So we know that from the eyes, we have the information coming out from the what we call optic nerve. Then we have two nuclei in the thalamus, and from thalamus we have visual cortex. So the information would be reflected to the visual cortex. And we know that how the information is being received from the environment is going to be reflected in the visual cortex in a way that we call, in visual cortex, we have a retinotopic map. So retinotopic map. Why do we have this? Because the information is to be spatially represented over the visual cortex in a way that, for example, if we go right in the visual field, there is a direction on the kind of visual cortex. If we go down in the visual field, there is that information is going to be on the upper part of the. So there is a something like representation or one by one kind of representation. Not exactly one by one, but there is a kind of a sort of kind of interaction between how these maps are really happening outside or how they are reflected to the visual cortex. Okay. There are some technical details in terms of, okay, if we cut this part of the visual cortex, if I cut this part, if I cut this part of visual cortex, if I cut these bundles, how the visual system is going to be affected. Okay. I'm not going to go to the details about that because, I mean, there are specific lessons in neurology, those who are in the kind of medical fields. That's one of the basic kind of information that students from the medical university, they learn in their courses. I'm not going to go to the details about that. I think it's not going to be that much critical for us right now. But just to give you a sense about how these things are being processed, there are specific devices that give you spotlights in different parts of your visual field. And when you see that spotlight, you need to just kind of push a button. Okay, that's how it is working. And over the time, they just put the spotlight in different parts of your peripheral visual system, and you just kind of keep on pushing the button, and then they make a map of the areas that you can see and the maps that areas of the visual field that you cannot see. For example, here, this map is showing what we call hemianopsia or hemianopia. It means one part of the visual field is basically blinded, so you cannot see that part. What is this? This is the blind spot. So even in a normal kind of person, if you do the testing on our eyes, in each of the eyes you have a blind spot. So there be the normal kind of, in the normal one, this part would be kind of bright, but you still have that blind spot in one piece, as you can see here, okay? And even based on the blind spot, you can see that it's the right map or the left map. So you can kind of detect that. But we can see what are the areas that are basically blinded when we cut these pieces from the visual stream of information. And people have different type of maps that are related to different parts of the system. For example, if you cut the pieces that are more towards the down part of the system, you can see that the upper part would be affected of the visual cortex because the retinotopic map is upside down. So the directions are in the opposite side. And that's the reason when you cut the upper bank, you have the problem in the lower part. Even it might be interesting that we can make some of these kind of visual, I should say probably visual hallucinations by stimulating visual cortex with TMS. So I can put TMS over your visual cortex. I can stimulate your visual cortex, and you start to see some moving dots or lines in the periphery around you, and we call them phosphene. So we can make phosphene in your visual field with directly stimulating your visual cortex. So that's what I've done for I mean, we can frequently do this in some of the cases, even to make a threshold to see what is the amount of power that we need for TMS to be able to make a phosphene on the visual cortex. But it's interesting that when a patient is telling you that they can see a phosphene, if you want to get sure that the phosphine is real, you can go up and do the stimulation again, and the patient should tell you that now they are seeing the phosphine on the lower part of the visual field. If they say that they are seeing that in the upper kind of area, it is a false one. So they're not telling you that the right one. The right one is when you go to the visual cortex, the down, you stimulate the lower part of the visual cortex, the phosphine that you see is going to be the upper side of the visual field that you have around you. That is an interesting directionality of the stimulation over the visual cortex. But technically, they have different names for them, something like bitemporal anopsia or hemianopsia or different types of kind of names that people are using. I'm not going to go to the details about that. It's not going to be that much helpful for us right now. It might be interesting that here in the visual kind of occipital lobe, we have what we call macular sparing. So those who are interested, they can think about why we have macular sparing here and why we have macular sparing in these graphs. So that's another thing I'm not going to discuss about right now. Okay. So to give you about kind of a better sense about the visual system, this is the kind of the figure that could give you a sense about what we have in the visual system. We have what we call V1, V2, V3, V4. So we have different layers of information that are being processed in the visual cortex, as you can see here in this figure. Okay, and let me just give you a better sense how these kind of pieces are representing the information. The information that we have in lateral geniculoid nucleus, which is this part, the thalamus part, is just in the level of dots. So the representation of environment around us is being processed in the receptive field and represented in lateral geniculoid nucleus as just simple dots. Then in the V1, we have mainly lines, and then orientation. Some levels of color would be perceived in the V1. And then in V4, we have three-dimensional shapes, we have curvatures, we have color. And then when we go to these parts, here is named as inferior temporal cortex, but you can name it as ventral temporal cortex as well, because the inferior parts are actually going to the down, it's going to be ventral part as well. So this one could be called ventral temporal cortex. Then in the ventral temporal cortex, we have objects and features. Okay, so that is how the information is being represented in different layers, from V1 to V2 to V3. So it goes through the kind of the process, but the image format is different in different representations in different layers. Okay, the other important part is that what we have seen before is something like this. We have kind of the environment visual field. The environment is reflected on the eyes, and then it has been reflected to the visual cortex. But it's also important for us to know that something like 10% of the information do not go through the lateral geniculate body or the thalamus. They go directly from the optic nerve to some other parts of the brain. So they do not go to the thalamus. So they go to other parts of the brain. So something like 10% of the information. Why? Why do we need this shortcut in the information? Let me just give you another photo here. Okay, so here what you can see. Other than just having the information in the lateral geniculate nucleus, LGN, which is a part of the thalamus, this is a part of the thalamus. This part of thalamus is receiving information, and it reflects or relay information to the primary visual cortex. So that is where we have the information. But otherwise, the information, a small piece of information, would go to other nuclei in thalamus, which is called pulvinar. Some of the as you can see here we have a path that is not going to the lateral geniculate nucleus. It goes to what we call superior colliculus. So we have two colliculus here, inferior colliculus and superior colliculus. The inferior colliculus, if you remember, was related to the auditory cortex. So in auditory information was basically going to the inferior colliculus. But the superior colliculus is receiving information directly from the eyes. And then from there, it could go to pulvinar, and then it could go directly to amygdala. There are some ideas that there might be even some pathways directly from optic nerve to amygdala. So there is a shortcut of information from the periphery to amygdala. And people believe that shortcut is designed basically for snakes, because we needed that information in a really fast way, going directly from the visual field to amygdala to be able to inform you about things that are dangerous in your environment, to be able to activate the fear sense in amygdala. So it happens to you that you see something in the environment, you still don't know what is this, because it is not still in your visual cortex. It is just going from your eyes and directly to your amygdala, and your amygdala would make a kind of fear response and make you fly or try to just go away from the source of threat. So that is how the system is working in a really efficient and rapid way. Otherwise, you would definitely be beaten by the snake. And that is the pathway that is being designed for that kind of processes to be able to make you really fast in responding to your environment. Okay, so we know that all the information is not going to the visual cortex. Some pieces of information would go to other pieces of the brain to make some other kind of responses. So there are some other kind of things that are happening here, as well as for example, in the superior colliculus, there is a pathway to hypothalamus to be able to arrange the level of, let's say, your metabolism based on the darkness in the environment. So it basically controls the level of circadian rhythms based on the kind of the illumination or the brightness that you have in the environment, the level of light that you have in the environment. So there are some other functions for that as well. So I'm not going to discuss about them right now. So this is another kind of photo which is showing exactly the same idea of that information is not directly going to the thalamus. It could go to the superior colliculus, and then from superior colliculus to other parts. And there are information that are not going directly to visual cortex, and they can go to other pieces without going to V1. So it might be interesting for you that sometimes we have damage to visual V1 in some of the disorders. And those people do not see the environment. The environment, they are blinded, so they cannot see anything in the environment. But because they have this pathway to the areas if you remember, we had two major pathways, dorsal pathway and ventral pathway. And dorsal pathway what was the name of dorsal pathway? We called that what pathway? Compared to the ventral, which was where pathway, and this one was what pathway. Okay, so we have some where information in the kind of areas here. How does it help? Some of these people, they have damage to V1. If you ask them to run in a maze or in a place that has different kind of obstacles and objects in the path, they would not hit the objects. They would just pass the objects. They would just go around the objects without hitting the objects. Why? Because they cannot see the objects. They cannot see the objects, but they can still find their path because they know that there are obstacles, because they know where the obstacles are. And the visual system is working hand to hand with the motor system that is here to just get sure you are not going to hit those objects in your environment while you are not seeing the objects. And that's a really complexity of this system because of this direct pathway. This is not going to the V1. It's just going directly to where areas. And there is no pathway to the kind of the ventral stream path. So you do not see them. You do not know what is happening outside. Okay. Some really simple kind of anatomical kind of descriptions about the areas are important. We have what we call lateral occipital cortex that I showed you in the brain automata atlas before today as well. We have an area that we call kind of it's basically in the middle temporal gyrus, but we call that kind of a motion area, so it is related to motion. And then we have an area which is called FFA. Again, an area we call PPA, FFA, V1, some of these areas. What is FFA? FFA is face fusiform area, fusiform face area. And PPA is place or parahippocampal place area. So it's a place area in the parahippocampal gyrus, is the face area. So these areas are specialized for detecting places and faces in the ventral pathway, which is the what pathway, not where pathway. This is the what pathway. So they are specialized to faces and places. So this is how you can see these areas. V1, V2, V3, V4, these areas are processing the information, and then these PPA and FFA. These are the really simplistic way of showing these areas. This is a more kind of advanced way of doing that. This is the inflated brain that I told you before. There are softwares that could make the brain inflated, and they can even make something like a surface from the brain. So they can just make from the entire cortical gyri and sulci, make a really flat surface. And that's how they try to flat them, to show you how these areas are connected actually. But sometimes it's really hard to see them when they are in the folding version of the brain under different sulci and gyri. So you can not see them really perfectly well. But when you make them flattened, then you can see how these areas are in relationship to each other, and how these kind of multiple levels of information are processing gradually there. But in reality, when we see them in the actual fMRI studies, they are a little bit messed up in terms of the exact borders of these activations. So we have mainly lateral occipital areas that are kind of detecting objects, generally any object. But face areas and place areas are related to fusiform face area and parahippocampal place area. But you can see that it's not a really clear line and distinction between these areas. They are a little bit overlapped, and based on the experiment that you do, they could be even more overlapped to each other. Okay, so that is the last piece of that part of the discussion today. Just to make a reminder for you, what we called usually V1 at the striate cortex, because this cortex is really multi-layer, so it has something like strias over the structure of the cortex. And then we name other parts of the visual cortex that are out of the striate cortex as extra striate cortex. So I mean, just a kind of a naming or taxonomy or naming agreement between people, but it's not that much critically important. So these are the kind of the pieces of information that we discuss about in terms of the general anatomy of this system. So we started with some kind of ideas about how the visual system is working. We discussed about the processing ways, and then we discussed about the general anatomy, and then we are going to go to the next level.